Hey everyone, this is Corbin from nxcplants, and today we're going to be talking about tissue culture. I'm going to start off by talking about what is tissue culture. So tissue culture is the process of regenerating plants from parts of that plant's tissue in a sterile environment. So this is applying growth hormones like cytokinins, which stimulate cell growth and differentiation and prevent stem cell differentiation, and auxins, which stimulate root development, vascular differentiation, and cell elongation to form those nice long cells that you see in the structures of plants. So in this process, a small piece of the plant tissue, be it a node, a root, a piece of stem, a leaf, whatever it may be, is taken from the plant and introduced into tissue culture in a sterile environment. So this is done on a media called MS media, and the media contains all of the necessary nutrients for the plants to grow. Your nitrogen, your phosphorus, your potassium, your calcium, your magnesium, your sulfur, as well as a whole host of other micronutrients like minerals and vitamins. So there's two main ways that tissue culture is done. It's done using either a meristem component of the plant, so this would be the top growth point of the plant, the cataphyll or what have you, or a node, an axillary bud. And this is sort of the best way to do tissue culture because it's almost like you're just growing the plant out as if you were to buy a node off Facebook Marketplace and grow the plant as you do. It's just being done in a sterile environment with the addition of some growth hormones to help it out. The most common way that tissue culture is done is through what's called a callus formation. So you might have seen this in some of those photos on Facebook where some of our intrepid anthurium friends are tissue culturing their plants. And it's the little sort of nasty looking growth, like a whole bunch of little bubbles coming from just like some spot on a piece of plant tissue. So the callus is induced using these previously mentioned growth hormones and growth regulators. And it's not known, but it's thought to be analogous or similar to a wound response in the plant that you might see in the wild. So if a plant is really severely damaged, it might induce callus formation so that the plant can continue to survive and it'll grow and live to see another day. One of the major side effects that's been recognized in the last 40, 50 years has been the presence of what's called somaclonal variation in tissue cultures. So somaclonal variation are mutations that happen to the genetics of the plant that causes them to present differently than the plant that had initially been introduced into tissue culture. So an example of what this might look like is these new round of tissue culture Thai constellations that have been coming out. Everyone's familiar with the Thai constellation. Nice round leaves with the beautiful speckling and odd sectoral cream color. But this most recent round, a lot have been cropping up with an additional layer of green on green variegation. Now, I don't know this to be a fact, but I theorize that the tissue culture labs didn't notice this when they were sending them out to the garden centers. You can pick them up at Home Depot. I found them at my local garden center. So these are things that are sort of sneaking by the quality control measures of whatever TC lab these companies are using. I think this because if the growers had noticed that there's a difference, they would have tried to sell it as something else, probably for a bit of a higher price. It's kind of just how the game works sometimes. But I say this is a great example of somaclonal variation because somebody put in a regular Thai constellation into TC, expected to get a regular Thai constellation out, and they got something that is mutated. So how are these mutations caused? These mutations are caused by three main mechanisms. The first is through transposons, movable elements within the DNA. So as part of a stress response, the plants have genes that will activate that move certain segments of their DNA to other segments, likely in the hopes of improving their chance at survival under a stressful situation. The other way is polyploidy, which is the duplication or deletion of chromosomes, which results in differential gene expression in the tissue cultured plant compared to the original plant. And the third way is single nucleotide polymorphisms, or SNPs, and these are single point mutations which occur at some point in the genome that causes a change in gene expression and therefore a change in how the plant appears. So how does tissue culture cause these mutations to occur? The first way is through the method used. As I mentioned previously, the difference between meristem culture and callus formation-based culture. Meristem-based culture is a more surefire way to get a plant that looks like the plant that you're culturing out of tissue culture. So it's commonly used when you really want to make sure that you're getting what you put in back out, and you don't necessarily care as much about the yield or getting as many plants as possible out of the culture. Tissue culture labs have realized that callus formation is associated with a much higher rate of mutation, so much so to the point that some tissue culture labs completely avoid the formation of a callus as part of their tissue culture process to reduce the number of mutations that are induced. So the damage caused when calluses form can create stress in the plant, and the stress and the rapid cell proliferation can lead to mutations. So think of mouth cancer or lung cancer in smokers. Those cells are being stressed and introduced to carcinogens, which causes gene mutations, cell proliferation. You get cancer. It's not the same, but it's a similar idea. And generally, the more disorganized a plant growth is, the more likely mutations are to occur. And because callus formation completely breaks down all of the natural tissues and the growth habit of the plants, this really disorganized growth can lead to a higher rate of mutations. The next way tissue culture can induce mutation is the time in culture and the number of subcultures. So a subculture is where you culture your plant, you break it apart, and then you put it back into culture and you rinse and repeat, and you get more plants out of it. Now, usually a good tissue culture lab will grow their culture for a certain amount of time, say a year, and then they'll throw away all of their cultures, they'll resample from their original mother plant to put back into culture to make sure that they're not getting much or any genetic drift. They also try to limit the number of subcultures, as I said, maybe a year, and then to toss out their plants. Conversely, plants can be maintained longer in tissue culture, and this will increase the number of mutations that appear. So this is how we get a lot of our Monstera spores. Obviously the ones that are grown from seed are grown from seed, but a lot of the tissue culture Monstera spores we get through this continual subculture. So the third way is the application of these plant growth regulators. So benzoadenine, or BA, is a commonly used cytokinin growth regulator in plant tissue culture, and studies have shown that an increasing level of BA used in the culture is directly associated with an increase in mutations in that same culture. Conversely, a lower level of BA used is associated with a lower level of mutation. And there's other synthetic hormones that are used in tissue culture as well, and many of these have been associated with polyploidy, so chromosomal duplication events or chromosomal deletion events, which change the amount of genetics that the plant has. So the fourth way that tissue culture can cause mutations in tissue cultured plants is by the proliferation rate. So the proliferation rate is how quickly the cells in tissue culture grow and how many little plantlets you get. So tissue culture can grow 30, 50, 100 plants in a year through the increased proliferation rate and subsequent subculture of the tissue culture plants. This is compared to if you were to cut a plant, like stem propagate a plant, you might be able to get 5, maybe 6 if it's a nice quickly growing plant, to grow out to a good little size. And I found a paper that said that the proliferation rate of about 4 per year was associated with a 5% mutation rate, which was considered acceptable by the researchers. But having 1 in 20 plants come out of your culture being mutated is still a pretty high number. Another paper that I found stated that their subcultures, so if they took a plant out of tissue culture, broke it up, put it back into subculture, they did that 5 times, they found a 1.3% mutation rate. So 1 in, I don't know, 80 or something. And the same paper found that after 11 subcultures, the mutation rate increased to 3.8%. So this is a pretty drastic increase and shows that continued subculture of the same culture of tissue cultured plants can drastically increase the number of mutations in that culture. So another way is through stressing the plant. So as I mentioned before, stressing the plant induces these transposable elements to move from one segment of the genome to another, thereby causing mutations. So this is a defense mechanism that's built into plants, and it's supposed to make them more resilient to whatever environmental stressor is stressing them out. But in tissue culture, it can lead to changes in the appearance of the plant. Another way that tissue culture can cause mutation is just through sort of natural selection. If you imagine you have two plants growing in the same flask next to each other, and one of them is mutated to have let's say a larger leaf blade and a higher growth rate. That mutated plant is now going to take over the original plants in those cultures because they grow faster, better, healthier, bigger. So if a grower isn't careful to select out these better growing but not desired plants, then you introduce these mutations into your plant population. And these mutants can also be selected by the culture environment. So the hormones that are used or the nutrients that are used might be more beneficial to a certain mutant that's appeared in the culture compared to the original one. And this can cause that plant to grow faster, again, faster, bigger, healthier, stronger, and outcompete the original plant that's in the culture. Finally, the tissue culture process can change the number of chromosomes that an anthurium has, or that a plant has. So plants come with a set number of chromosomes, usually 2 pairs of however many. Anthurium, on average, they'll have 2 sets of 30. But tissue culture can cause them to have 3 sets of 30 or 4 sets of 30. Or they can cause them to have 3 sets of some chromosomes and 2 sets of others and 1 set of others, just causing changes in the number of chromosomes of the plants. And again, this is a source of mutation. If you're changing your number of chromosomes, your plant is going to look and behave differently. Tissue cultured plants that have more chromosomes are more likely to have this type of polyploidy mutation occur while they're being tissue cultured. So something like sugarcane that I found in a paper has a high number of chromosomes, in the 80s or 90s, I believe. They have these chromosomal mutation rates at much higher rates of other plants that have lower chromosome numbers. So anthurium generally are on the lower side of chromosome number with their 30. So I'm not sure that this would really be a driving factor behind anthuriums specifically mutating in tissue culture, but it is a factor nonetheless, as there are many anthuriums which have strange number of chromosomes, extra chromosomes, B chromosomes, or just like more than 30. In tissue culture and out of tissue culture, plants mutate at a pretty regular rate of around 1 in 100,000 or a million genes. The average plant has between 20,000 and 40,000 genes. I'm assuming anthurium are similar. I couldn't find anything specific for the whole genome of anthurium. But if you do know, please drop a link down in the comments. With these numbers, your average anthurium that you're maybe going to grow from seed or tissue culture, you have a mutation rate of 1 in 2 to 1 in 5 plants. That being said, not all of these mutations are going to be mutations that allow the plant to survive. In fact, most of them will outright kill the plant. Now, researchers have found that in tissue culture, a mutation rate of a culture of between 15 and 20% is pretty common, but I've also seen numbers as low as 1.3%. So again, the science isn't really settled on the exact numbers of mutations and their sources of plants coming out of tissue culture. And the final really important factor in tissue culture is trust. You have to be able to trust that the lab you're buying from is using the plant that they said they were, using good tissue culture practices, not over subculturing their plants to get as many out as possible, not blasting them with growth hormones to make them grow as quickly as possible. All of these things can induce mutations. And yeah, super important that they are using the plant that they say they're using, because your US grower who commissions a tissue culture from a lab in Thailand, who then subcontracts that out to other people wherever they may be in the world, each step along the way you have to be confident that the plant that they're saying they're using is what they're using, and that they're using their best tissue culture practices to grow those tissue culture plants. The field of somaclonal variation in tissue cultured plants is definitely one that has been around for quite a while, but it's mostly being used for agricultural crops. Bananas, sugarcane, wheat, soy, corn. There's not a lot of research into somaclonal mutations in foliage or floriculture plants, anthuriums being one of those. So definitely we would need more research to be able to definitively say this is the way to TC anthuriums, this is the way to not TC anthurium. But that being said, tissue culture is not bad. I didn't come out here to talk bad about tissue culture. I think tissue culture is a really incredible way of making these inaccessible plants accessible. If you think about the Kunzo white monster, that guy a couple years ago was going for thousands and thousands and thousands of dollars per plant, and now you can buy them at a TC for $40. Like, I think that is a really great achievement, because it not only reduces the pressure on the growers, it makes the plants more accessible for those of us in the hobby who might not be willing to drop thousands of dollars left, right, and center on whatever new hot thing is out. Beyond that, for anthurium, if you have a really excellent specimen, say like the circus peanuts or the Delta Force, these plants being tissue cultured, again, reduces the cost, reduces the pressure on the people who are growing the plants to divide them, and it makes these stunning plants that are part of our sort of shared culture and shared love in the anthurium hobby, and that are so important to that. It makes them more accessible so that everyone can enjoy growing them. But because tissue culture is, in theory, a clone of the plants that are being tissue cultured, anthurium, in my opinion, almost lose a little bit of their magic. So yes, tissue culture for one-of-a-kind specimens like the Delta Force or the circus peanuts is great, because these are singular one-off hybrids that have to be divided by stem, and they can't be recreated through seed in any other way. And even if you find a plant that like looks better to you or to lots of people, it's still not the circus peanuts or the Delta Force. And this a little bit falls apart when you're talking about maybe pure species or species that are grown from seed, because in my opinion, there is always a nicer version of the plant out there. Think about the variability in papillolaminum. If someone was to pick the RL, great example, as the nicest anthurium on the planet, you know, maybe I would even have a hard time disagreeing with them. But if they were to pick that as their plant to tissue culture and everybody has an RL, it's not really as special or prized anymore. Yes, it's really nice, but it's no longer uniquely nice. And now someone else in their collection who has, say, an RL self or a Lagogatun ecotype of pap that has different or more extreme characteristics, now that might be the new gold standard of the nicest pap in the world. So tissue culture sort of, it levels the playing field in both positive and negative ways. The positive being that it makes all these plants much more accessible, but it also removes the mystique and splendor of having an exceptional specimen that is, you know, unique to you, or maybe unique to you and a handful of friends. So one example of this is the Ace of Spades, and I have two, so let me pull them out. So this is my seed-grown Ace of Spades from Tazula. You can see it's got a really stunning horseshoe sinus, nice dark, like black velvet color, and it's got a little bit of pink in the veins. It's more of like a burgundy. This specimen is unique because it was seed grown by Tzula, selected for its characteristics and its traits, and sold by Tzula. So compare that to the tissue cultured ace of spades. At least I think it's tissue cultured. They almost have a look to them. I didn't buy it out of tissue culture. I don't know if it was out of tissue culture, but SKG did, and Tzula did do a large round of tissue cultured ace of spades quite a while ago, and I think that many of the ace of spades that are out in circulation today are derived from these tissue culture plants. So they do have a slightly different appearance. If I'm being honest, it's not quite as charcoal black as the seed-grown one. The sinus overlaps as opposed to having a nice horseshoe appearance. Might be able to see it better on this leaf here. It has an overlapping sinus versus the open horseshoe of this one here. It has more of a pale colored vein as opposed to the rich maroon purple of the seed-grown ace of spades. So this is an example of where the Tzula ace of spades that I'm holding, the smaller one, is a stunning plant. It's like almost black. It's got an incredible shape, teardrop shape, with very cute lobes and really nice minimal veins. But the specimen that was seed grown is just a little bit nicer in every way, subjectively, to me. It might not be the case for you, but for me, I think that the seed-grown ace of spades is just a little bit nicer in every way. But ultimately, it just comes down to your personal preference. If you prefer the look of whatever the TC plant is, be it the RL, the Delta Force, or you might prefer a seed-grown specimen that has a little bit more of an extreme characteristic, or you might just want the tissue culture one because it's cheap and easily accessible. Those are all the reasons that tissue culture is great. But that being said, you should always have a little bit of skepticism when you're buying plants, no matter where they're being sourced from, and making sure that first and foremost, you're trusting the seller. I will post all of my sources for this video down in the description. If you have any questions or any ideas for another deep dive research topic that you'd like to see me presenting, then please feel free to leave a comment. Thank you so much for watching. Have a good day. Bye.